These are AP Biology notes on cellular respiration and fermentation. This first set is about the overall need for respiration and respiration as a redox, series of redox reactions. So the reason we need cellular respiration is to provide energy for the cells to do whatever work they need to do. Energy is always required from some kind of outside source. And if you're a plant, of course, then sunlight provides that ultimate source of energy. But if you're an animal, generally speaking, you get your energy by eating plants or other organisms that eat plants, such as the chimpanzee. And remember that energy flows into an ecosystem and flows through the ecosystem, enters the sunlight, leaves as heat, whereas matter is recycled within the ecosystem. Photosynthesis, of course, produces oxygen as a byproduct and organic molecules as the main product of that process. And those organic molecules and the oxygen are both used in cellular respiration. We're going to use the energy that is stored in organic molecules like sugars to make more ATP or regenerate ATP, and that's what powers the work of the cell and ultimately of the organism itself. So here we have light energy flowing through the ecosystem and leaving as heat, of course. It is used by photosynthesis to produce those organic molecules and oxygen, which are used by the mitochondria to do cellular respiration, release carbon dioxide and water that are the reactants for photosynthesis, and as a result of respiration providing ATP, which powers cellular work. The excess energy, of course, is lost as heat as the energy flows through the ecosystem. So what we're talking about here is a series of catabolic reactions. Catabolic means to tear down or break down, and so what we're doing in the process of cellular respiration is oxidizing organic fuels through a series of catabolic pathways. And there are several different things that are important to that. First of all, then we're going to produce energy. This is an exergonic series of reactions. There are other methods besides cellular respiration of providing some ATP, including fermentation, which partially breaks down the sugars. It occurs without oxygen present. Aerobic respiration consumes those molecules plus oxygen, and that yields ultimately more ATP than you get from fermentation or anaerobic respiration. So we have and there's also other anaerobic respiration that can follow similar pathways using compounds other than oxygen to break down the organic molecules. The process, the overall process of cellular respiration includes aerobic respiration and anaerobic respiration, but when we use the term cellular respiration, we're generally referring to the aerobic respiration, the entire process. So when you look at, of course, the overall reaction here, realize that we're looking at glucose, which is one of the compounds used to provide energy when we look at the overall equation here, but realize, of course, that there are other molecules as well that can provide energy through the same or similar pathways. Now, the transfer of electrons, of course, is going to be something that's paramount of paramount importance to this entire process. And what we're talking about here is oxidation and reduction. We're going to transfer electrons from a source to a recipient and use that process to power the reactions that are involved in breaking down the glucose and other molecules to release or synthesize ATP. In redox, we are transferring those electrons, and those are called oxidation reduction reactions. Okay, so the oxidized substance is the substance that loses electrons, and the reduced substance is the substance that gains electrons. We're not going to worry too much about the chemical basis for this as far as going through all the chemistry of this. In biology, we're more concerned with the overall reaction. So we're talking about transferring electrons from one substance to another. In the case of sodium and chlorine, sodium becomes oxidized and loses an electron to become the sodium ion, and the chlorine gains an electron and becomes reduced to become the chloride ion. So we can look at a generic equation here showing the same kind of thing. We're just transferring an electron from one substance to another. The one that loses the electron, of course, becomes oxidized, and the one that gains the electron becomes reduced. So we're talking about reducing and oxidizing agents, okay? Sometimes, especially in biological systems, you have a redox reaction that doesn't actually involve the change in electron, the presence of electron in one, or transfer of electron from one substance to another, but a change in the sharing characteristics. So if you look here at the oxidation of methane, okay, methane is going to be the reducing agent, and it's going to lose some energy, and the oxygen is going to be the oxidizing energy agent. And so what we have here, rather than a transfer of electrons, we have a change in the electron sharing. Since oxygen is so much more electronegative than either carbon or hydrogen, the electrons in those covalent bonds are going to end up being closer to the oxygen atoms in both of these cases, and that's going to create, of course, some polarity within the molecules, but then it's also going to mean that this is what's going to be the oxidized form here, and this is the reduced form here. Notice that the electrons were not transferred, they're just the sharing characteristics were changed. In respiration, what happens, the fuel, like glucose, is oxidized and the oxygen is reduced. And so we show the reduction there by the addition of the hydrogen to the oxygen and the loss of hydrogen from the from the glucose to produce the carbon dioxide. In respiration, all this process happens in a series of steps. Okay, and so we're going to use several different organic compounds in the process, and we're going to use some various molecules like NAD+, which is a coenzyme, as agents to receive or transfer the electrons from one place to another. NAD+ is an electron acceptor, and so it's going to function as an oxidizing agent during this particular process. And then the reduced form, NADH, is going to represent stored energy that is used to synthesize ATP in the final stage of the process. Here we have NAD+, which is nicotinamide adenine dinucleotide. And you see here's the nicotinamide part, here's the adenine nucleotide. And it makes a double nucleotide here. And so what we're going to do is we're going to take a hydrogen from the food and add it here to the nicotinamide to make NADH rather than NAD+, and that's going to remove that hydrogen from this hydroxyl group and produce a carbonyl group on the molecule. The NADH is going to then pass those electrons on to the electron transport chain, and that's going to pass electrons through a series of steps to give a stepwise release of energy along the way rather than a huge explosive reaction in the end. And then the energy that is produced here is going to regenerate ATP at the end of the process. So here we have, if we had the uncontrolled reaction, of course, going from water to hydrogen and oxygen here, the free energy is indicated by G. But what we have in this particular process, the uncontrolled reaction, is an explosive release of energy. Whereas if we go in cellular respiration from hydrogen and oxygen to water in a stepwise fashion, what we have here is a controlled release of energy, and that is used to synthesize ATP, which then the cell can use for its processes. It's obviously a whole lot safer for the cell, as well as being a more controlled reaction. So let's just look at the overall process of what's going to happen. We're using glucose as the main molecule. There are other pathways as well, which we'll talk about later, but there are three main stages in the entire cellular respiration process. There's glycolysis, which is going to break down glucose into two molecules of pyruvate. There's the citric acid cycle, which is going to complete breaking down glucose and releasing the carbon dioxide. And then we have the oxidative phosphorylation process. That is where most of the ATP is produced. Here we have the mitochondrion, where in and near where these reactions occur. Glycolysis, of course, the first step happens in the cytosol or in the cytoplasm outside there. It produces pyruvate and some electrons that are carried by NADH there, the reduced form. It produces a little bit of ATP by means of substrate level phosphorylation. The next stage is the citric acid cycle after the pyruvate is oxidized to acetyl-CoA. Then it enters the citric acid cycle, where there are more NADH and some FADH2 that's produced, a little more ATP, again by substrate level phosphorylation. And then finally, we have the oxidative phosphorylation in the electron transport chain and chemiosmosis that produces the major amount of ATP using the electrons carried via NADH from glycolysis and the citric acid cycle. The main process that generates most of the ATP is called oxidative phosphorylation, and it's powered by redox reactions. It's about 90% of all the ATP that is generated for this process. There is a little bit of ATP produced in glycolysis and also in the citric acid cycle by means of substrate level phosphorylation, but most of the ATP is going to come from the oxidative phosphorylation and chemiosmosis process at the end. Here we have what happens with a substrate level phosphorylation. We have a substrate here that has a phosphate on it, and there's an enzyme that catalyzes the reaction to remove the phosphate from the phosphorylated substrate and add it to ATP to make ADP, producing a product that is not phosphorylated plus our ATP.